All right, here we go. I think this is recording, yeah? All right, here I go. I've got about, what, 25 minutes before I have to wrap up, yeah? So I will try to be lively. Of course, you guys know me by now, huh? Sorry about that. You might as well ask me to be a different person. All right, so, and yet I will try to be that different person right now, before your very eyes, by being rapid, okay, instead of long-winded, like I usually am. All right, so microbiology concerns itself with eukaryotes of all kingdoms, yeah? We're going to certainly spend more of our time working with bacteria than we will spend working with eukaryotic cells. But eukaryotic cells can still be pathogens. Animals can be parasitic. A tapeworm is an animal and is macroscopic, yet it's a parasite. We care about it in microbiology. Fungi can be pathogenic. Ringworm is a pathogen, and it's fungus, yeah? Likewise, malaria is a deadly parasite, yeah? It's an intracellular parasite, and it's a protist. So it's a eukaryotic cell that is causing serious human disease. It's one of the deadliest diseases worldwide. So what else do I have to say? Well, this is something I said just a few minutes ago, yeah? Penicillin blocks the action of enzymes that makes bridges in peptidoglycan. And why do we care about that? Well, because we don't have peptidoglycan, and if prokaryotic cells do, then that is a difference that we can exploit, yeah? It's an exploitable target, an ideal drug target, yeah? Well, next question, who found that target? Alexander Fleming? He was the one who discovered penicillin, but who made penicillin? Penicillium notatum, right? A fungus. And why did the fungus make penicillin, right? So how long has penicillin been around, the drug? Yeah, so how do we answer that question? We can say since 1928 when it was discovered, that's how long people have had the chance to use it. But these guys have been using it for millennia or millions of years or hundreds of millions of years, yeah? All right. Well, what do bacteria do? They just sit there and say, oh, kill me, kill me. No, there's an evolutionary arms race, right? Why did penicillin make it? Why did penicillin make penicillin? Should I rephrase that? Why did Penicillium notatum, this critter here, this fungus, make the chemical penicillin that is this chemical that's messing up the enzymes that help bacteria make peptidoglycan? Defense, self-defense? What do you think? I would say that penicillin is an offensive weapon, but it is certainly something that will help penicillium fungi in their war against bacteria. They are evolutionary competitors. They compete for food in the bottom of my fridge, and their mutual ancestors were competing out in the wild long before my fridge ever was filled with the vegetables that I just bought, yeah? Does that make sense? And what about bacteria? Sometime, tens or hundreds of millions of years ago, Penicillium notatum fungus made a chemical that could give Penicillium fungi an advantage. If you were bacteria, what would you do? Just sit there? Try to have a defense against it. What do we call a defense against penicillium? That's antibiotic resistance, yeah? When did antibiotic resistance show up? Just a few years ago? Maybe it just became a problem a few years ago, but it didn't just show up a few years ago. Does that make sense? It didn't show up evolutionarily a few years ago. We've been By natural selection, though, we've been enriching for, or by selection, when we take antibiotics, we enrich for resistant microbes, don't we? Right? But which came first, the drug or the resistance? Well, the drug came first 100 million years ago. The resistance came 95 million years ago, and then we started using it 50 years ago, yeah? Or 60 or 70 years ago now, yeah? Does that make sense? Okay. And then what happened 50 or 60 or 70 years ago, or excuse me, 40 years ago or 20 years ago, is that suddenly we had a lot more resistant microbes than we did before. Does that make sense? But the genes in bacteria for resistance pre-existed our use of antibiotics. Shall I say that again? The genes for resistance in bacteria pre-existed our use of antibiotics. The thing that's making antibiotic resistance a problem is that we are selecting for resistant microbes by the process or a special process of natural selection. Guys with me there? Okay. Moving on. So let's look at the eukaryotic cell. Why do we care? Again, knowing the differences between prokaryotic and eukaryotic cells, right, allows us to suss out the differences when we're targeting prokaryotes, yeah? So knowing the differences is a good thing. Eukaryotic cells have many structures in common with prokaryotes, yeah? The glycocalyx, the cell wall, plasma membrane, ribosome, cytoplasm, flagella, genome. These are all things that both prokaryotes and eukaryotes have, yeah? However, many of these are common in name and in function, but not in structure. Any idea what I'm talking about there? So what name, right? Glycocalyx, cell wall. Do humans have cell walls? Do eukaryotes have cell walls? Yes. Give me a eukaryote that has a cell wall. A plant. Plants have cell walls. How many people know what plant cell walls are made out of? Cellulose. How many people can I get an, oh yeah, right? How many people remember, they couldn't think of it, but now that it's been mentioned, they're like, oh yeah, right? So for me, my response is, can I get an, oh yeah? So can I get an, oh yeah? Oh yeah, okay, good. All right, so hopefully you remember from a previous class, someone in class has reminded you that plants have cell walls. Bacteria have cell walls, common in name. What does a bacterial cell wall do? Protects the cell, provides structure, prevents osmotic lysis. Yeah? What does a plant cell wall do? Protects the plant cell, provides structure. Yeah? Similar name, similar function, but not similar in structural composition. Yeah? You understand? Because a bacterial cell wall is made out of what? Peptidoglycan, as we just learned. And a plant cell wall, cellulose, a different structure. Does that make sense? Different. No, don't stop recording. So maybe I'll change this word, but not chemical composition. Is that better? That's what I mean there. Is that clear? Okay. All right, moving on. And you know what? I should probably save that change just so that change is saved. Okay. Ah, fiddlesticks. There we go. All right, so let's look at these. First, the glycocalyx. And again, we're gonna survey our organelles in the same way we going from the outside of the cell to the inside. Yeah, the glycocalyx is the outside sugar coating of a cell. The glycocalyx in bacteria is important. Yeah, and it determines pathogenicity, and it can allow biofilms. Well, we have a mucus layer on the inside of our intestines, yeah, in one of our mucous membranes, yeah. That mucus is water and polysaccharides, carbohydrates. So it is effectively a glycocalx, right? And if you remember your microvilli, right? Those are the brush border on your villi of your, yeah. So if you're going like this, you're showing me that you understand the structure of your intestines, maximizing surface area on the inside for absorption. Well, those microvilli do a lot to maximize the surface area of the villi themselves, and they have a glycocalyx, a mucous membrane, or a mucus layer rather, that protects them and yet allows the diffusion of nutrients. Okay, so cell wall. Eukaryotes don't make peptidoglycan, but they do make cell walls. Fungi have cell walls. Their cell walls are made out of chitin. And we talked about chitin as a modified glucose polymer. Likewise, plant cell walls are made out of cellulose, which is a glucose polymer that has alternating upside down, right side up glucose subunits that allows hydrogen bonding. There's also lignin in there, so it's lignocellulose. And then diatoms are photosynthetic protists. These are also eukaryotes. They have silicon cell walls. Their cell walls effectively are made of silicon dioxide, the same stuff that glass is made out of. And they look like glass, don't they? Aren't they cool looking? Aren't they pretty? Right, so diatoms are photosynthetic, and they have cell walls. We have glass cell walls, plant cell walls of cellulose, and chitin cell walls in fungi. Right? And there's more plant cell walls. All right, what about ribosomes? Do prokaryotes have ribosomes? Yes. Do eukaryotes have ribosomes? Yes. So every living thing has ribosomes, but again, the ribosomes aren't the same. They have 70S ribosomes. We have 80S ribosomes. And again, we have our wonderful ribosomal math where our 80S ribosome is made out of a 60S big subunit and a 40S small subunit, right? Okay. All right. And again, because bacterial ribosomes are different, they are a target for antibiotics. So everyone, please take a look at this picture. Yeah? You see this line here, this arrow going in here? What kind of ribosome is this? Is this a prokaryotic or a eukaryotic ribosome? It's prokaryotic. How do we know? It's 70S, right? And so this arrow says, can you guys read this? It's probably a little small. You read this, it says viomycin and capreomycin site. What does that mean? How many people have heard of viomycin? I have not. How many people have heard of capreomycin? I haven't heard of that one either. But what do you think those are? Antibiotics, right? How did you know that they were antibiotics? You've never heard of them before. The mycin suffix, yeah? What does that sound like to you? Erythromycin, streptomycin, those are antibiotics you have heard of. Does that make sense? All right, so these are antibiotics. And what's this arrow? This says site. What is a site? It's a place, yeah? Well, what does the ribosome do? Do you remember? It's a site to make proteins. And we saw how it does it. It takes a messenger RNA, and the messenger RNA runs through, and it reads the messenger RNA, and it makes the proteins, and the proteins come out of its head. Yeah? So, in between here, what's supposed to go here? That's where the messenger RNA is supposed to go, right? But what if viomycin lands here? What do you think would happen? It would block it, right? Maybe it could clog the ribosome. Do you understand? And if it clogs the ribosome, then what? You can't make proteins. And if you can't make proteins, then what? You can't make squat. You can't make your cell wall. You can't make your cell membranes. And what happens? You die, yeah? You can't make a cell and you die. So if you can basically, effectively, these antibiotics are acting like a wrench sticking in some gears, right? A ribosome is a precision machine. It's machinery that basically acts like little gears, and it pulls a little ribbon of messenger RNA through this complex device, yeah? What does the antibiotic do? It gets in there and messes it up, right? Why am I spending so much time with this? Because this mechanism called inhibition is something you're going to see a lot, right? Let's go back to penicillin. How does penicillin work? Well, this is the protein. What job does this protein have? We know this is a protein. We've seen it a couple times today, yeah? What does it do? Makes the cell wall. What's this doing? It's getting up in the protein's biz, yeah? It's impeding it, yeah? And so if this penicillin is getting all up in this protein's business, then what happens? You can't make a cell wall. And if you can't make a cell wall, what happens? You die. Does that make sense? Right? So, again, proteins are precision machines. Precision machines can be messed up. That messing up we technically call inhibition, but you can see it here, right? Is this going to mess up our ribosomes? I would be more scared of this messing up my ribosomes than I would be of penicillin messing up my ability to make peptidoglycan, right? But I can comfort myself that these ribosomes are different. Does that make sense? Because they're different, they can be a target for antibiotics, right? I would be less excited about taking an antibiotic that targeted a ribosome than one that targeted peptidoglycan. But we can find these things out scientifically by doing experiments, right? And if somebody takes it and they don't die, then maybe somebody else would be more willing to take it. Does that make sense? Okay. All right, so much for that. That's an important concept. If it flew over your head, you'll get another crack at it, okay? Because we'll be talking about it again. All right. Cytoplasm is, here's your poetry for the day. Cytoplasm is cytoplasm. Okay, so not a whole lot of difference between, it's fluid inside a cell. Okay. Genome. We already know the term genome. You should know this term. All the DNA and therefore all the genes of any cell, individual, or species. The genome of a prokaryote is in the nucleoid region. It's one chromosome, circular chromosome. What about the chromosomes of a eukaryotic cell? How many chromosomes do you have? How many people know how many chromosomes people have? Most of you. How many? 23 pairs, therefore 46, yeah? So bacteria have generally one piece of DNA, one chromosome, not in a nucleus. Humans have 46 or 23 pairs for a total of 46 chromosomes in a nucleus, yeah? The chromosomes of bacteria are circular. That is a single loop of DNA. We have lines of DNA, 46 linear pieces of DNA. Yeah, and so here they are. Those are the chromosomes we might be used to seeing when we learn about mitosis and meiosis. There's 46 of them, and there's 23 kinds. We've got about 17 to 25,000 genes. That means about 1,000 genes per pop. All right, so why do we see globular X-shaped things? Well, because their DNA is wrapped with proteins, right? Why don't we see a double helix here? Well, because the double helix is wrapped with a bunch of proteins. Got it? Those proteins, the most basic proteins, are what are called histones, and those act like little spools on which DNA can be wound. Okay. So much for the organelles that prokaryotes and eukaryotes have in common. Now let's look at the ones that are unique to eukaryotes. I'll try to go fast on these because most of these I expect you guys already covered in your human bio and your physio classes, yeah? So you've heard about these twice. I'll try to go fast. Nucleus, that's where the DNA is, yeah? Okay. The endoplasmic reticulum, do you guys remember this one? This is the network of membranes. You can have rough ER that has ribosomes on it for proteins that are exported from multicellular cells. Smooth ER has enzymes on it for detoxification and manufacture of phospholipids. Okay, the Golgi complex modifies proteins. You've got vesicles that can land on the sort of cis side and go to the trans side. Inside, those vesicles are little bubbles of proteins. Those little bubbles of proteins can be modified as they mature in the Golgi. They can be modified and turning them with carbohydrates and lipids to make lipoproteins, glycoproteins. Does that make sense? After you've made your proteins, you can modify them with non-protein aftermarket modifications in the same way you might modify your car with fancy wheels or the spoiler in the back or the plush seats. Yeah? Okay. All right. That's what the Golgi complex does. The Golgi complex can also make lysosomes. Lysosomes are modified vesicles. They're filled with digestive enzymes. Yeah? Those enzymes exist at a different pH, generally a more acidic pH than the rest of the inside of the cell. So these act very analogous to and very similar to the way your stomach acts, right? Insofar as organelles are little organs, the lysosome is the stomach, yeah? Hopefully that's familiar to you. And we can see how lysosomes work when we do something called phagocytosis, yeah? A particle of food is engulfed by phagocytosis. The bubble of that food vacuole is fused with a lysosome, and the enzymes in the lysosome will break down that food particle into little pieces. Yeah? 10-minute warning. Thank you. Thank you. All right. Could you give me a 5-minute warning too, please? All right. So, why do we care? Well, this is sort of theorized little food particle. Little food particle could be a pathogenic bacterium, yeah? And pathogenic bacteriums, when they get swallowed up by food vacuoles and then fused with a lysosome, what's going to happen to that bacterium? It's going to get digested, adios bacterium, right? And if that were the case, what kind of cell would you say this probably was? Say it again. White blood cell, yeah? Macrophages, monocytes, I think is another term. Those are kinds of white blood cells that do phagocytosis, yeah? Well, if you were a pathogen, you might not be surprised to see that some pathogens exploit eukaryotic endocytosis to infect cells or avoid destruction by lysosomal fusion, right? Again, there's an arms race out there. And these bacteria, good parasites, don't just say, kill me, kill me, kill me. That's fine. They have mechanisms. If they're going to be effective pathogens, they had darn well better have effective ways to subvert, to get around the human immune system. You understand? Well, some of them can, once they're swallowed up, can bust out of that vesicle before that vesicle fuses. Others have a waxy coat that makes them, once they do fuse with that lysosome, the acidity and enzymes of the lysosome can't really break them down because they're covered with this waxy coat. And so on and so forth. I'll show you guys. In fact, maybe I'll show you pretty soon, but yeah, I don't have time right now. Okay, so this organelle manufactures proteins. Please, everybody get this right. Ribosome, nicely done. Okay. Okay, mitochondria. Mitochondria provide about 90% of ATP for eukaryotic cells. Hopefully you've seen mitochondria before. If you have, you might have seen double membranes, yeah? An outer membrane and an inner membrane, with the inner membrane all folded up. Those inner folds are called cristae or cristae. I don't really know how to pronounce them, but those little folds are special, and they have a special name, and they happen to the inner fold. Why do you think it's all folded up? More surface area. So there must be something special about that membrane. What else do we know about mitochondria? Well, they have their own DNA. If we look at inside what's called the matrix, this blue part of this diagram is called the matrix of the mitochondria. Inside, there's little loops of circular DNA. Circular DNA, where do we find that? Bacteria, yeah? There are also ribosomes. If we look at the ribosomes inside the mitochondria, we see that there's 70S ribosomes. What do we think there? Like bacteria, huh? Crazy, isn't that? So, plant cells have some distinctive organelles that animal cells don't have. A central vacuole is usually filled with fluid. Let's see, they also have chloroplasts. Chloroplasts are little organelles that do photosynthesis. They have chlorophyll, that's the green pigment that captures the light. They also have their own DNA inside and their own ribosomes inside. And again, like mitochondria, their ribosomes are 70S. Why do you think, how many people know why they have their own, mitochondria and chloroplasts have their own DNA and their own ribosomes? Because they don't need oxygen? What do you think? So actually, as we'll learn, mitochondria use oxygen to provide 90% of our ATP. Without mitochondria in our cells, our ability to make ATP for energy goes down by 90%. Do you guys know why they have their own DNA and their own ribosomes? They can actually reproduce on their own. The cell will send out a signal, and then those they're not exactly prokaryotes. Those mitochondria and those chloroplasts will reproduce by binary fission inside your cells. If you exercise, if you do endurance exercise, you've got mitochondria in your muscles to provide energy for your muscles to do that exercise. And if you exercise regularly, you'll suddenly end up with more mitochondria than you had before you started exercising. And those mitochondria would have increased their numbers by doing binary fission. So, as it turns out, the reason is because of something called the endosymbiotic theory. This was first put out by a lady named Lynn Margulis. And Lynn Margulis was a professor at UMass Amherst. And she was also married to Carl Sagan. Do you guys know who Carl Sagan is? Billions and billions. Anyone see Cosmos with Neil deGrasse Tyson? Carl Sagan was an important mentor to Neil deGrasse Tyson back in the day. Anyway, and she's from UMass, so she's local, I guess. Represent and all that. So she put out the idea that eukaryotes came from what's called an endosymbiosis, where mitochondria and, for plants, chloroplasts got swallowed up, kind of like we might swallow up bacteria here, yeah? And instead of fusing with a lysosome, kind of like some of these guys can sort of dodge that, they instead started forming a partnership, yeah? Do we benefit from having mitochondria? Yeah, our ability to have energy has gone up by tenfold, right? we now can make 10 times more energy for our cells because we have mitochondria helping us out. Do they benefit? Well, certainly the evidence suggests all eukaryotes, right? All eukaryotes have mitochondria in their cells. No eukaryotes exist without mitochondria. And for every eukaryotic cell, there's dozens or hundreds of mitochondria. So their numbers are great. Would you not agree? Evolutionarily, the mitochondria has been successful. Right? Okay. Lastly, the cytoskeleton. Cytoskeleton, this is not in your text, but it's stuff I want you to know. This is in eukaryotes have a cytoskeleton. Some archaea have cytoskeletons, but no bacteria do. Okay, inside there's microtubules, microfilaments, intermediate filaments, but what they sort of do is provide a network, a structure in which sort of a highway system where we can see things moving around, yeah? So microtubules form cilia, flagella. Flagella are made out of bundles of microtubules. And sperm have flagella, yeah? Don't bacteria have flagella? How are the flagella of sperm related to the bacteria of flagella of bacteria? Well, they're related by analogy, not by homology, right? They are similar in name and in function, but not in chemical composition. Does that make sense? And they don't have a common evolutionary origin. Okay, so briefly, this is a microtubule. Can you see the nucleus of this cell? Yeah? Can you see the body of the cell? Can you see all the little green filaments? Aren't they? They're kind of cool looking, yeah? So these filaments are basically a highway network. So I want to show a video, and then we'll probably, I'll have to call it a day. Go to the internets. Sorry about the delay. So I'm at 1 minute. Is that right? And I'm about to show a 3-minute video. I'm going to go a little over, but you guys, I promise, will have at least 30 minutes. Okay? For lunch. Okay, YouTube. So we're going to go kinesin on microtubule. So I think this is
the motor protein kinesin is a dimer with 2 identical motor heads. Motor protein kinesin is a dimer with 2 identical motor heads. Each protein kinesin.
So what is this thing we're looking at?
The motor protein kinesin.
What is it now? It's a motor protein. And what's its name? Kinesin, and it's a dimer, 1, 2, right? So it's a protein that has 2 subunits, yeah?
Is a dimer with 2 identical motor heads. Each head consists of a catalytic core and a neck linker. In the cell, kinesins pull organelles along microtubule tracks. The organelle attaches to the other end of the long coiled coil that holds the 2 motor heads together. The organelle is not shown here. In solution, both kinesin heads contain tightly bound ADP and move randomly, driven by Brownian motion. When one of the 2 kinesin heads encounters a microtubule, it binds tightly. Microtubule binding causes ADP to be released from the attached head. ATP then rapidly enters the empty nucleotide binding site. This nucleotide exchange triggers the neck linker to zipper onto the catalytic core. This action throws the second head forward and brings it near the next binding site on the microtubule. The attached trailing head hydrolyzes the ATP and releases phosphate. As the neck linker unzippers from the trailing head, the leading head exchanges its nucleotide and zippers its neck linker onto the catalytic core, and the cycle repeats. In this way, kinesin dimers move processively, step by step, along the microtubule.
All right, so what is this thing? What is this thing that we're looking at called? This is kinesin, and kinesin is what? It is a motor protein, yeah? So this is a protein, yeah? What's it doing? Is it making ATP? It's using ATP. And what's it using the ATP to do?
Unzippers from the trailing head. The kinesin dimers move processively, step by step, along the microtubule.
So there's several things I want you to take home from this. And I found this by going to YouTube and saying kinesin walking on a microtubule, yeah? But basically what we see is A, proteins are sophisticated, right? This is a molecule. Is it alive? Is it alive? Raise your hand. It's alive. Raise your hand. It's not alive. So majority people voted yes, it's alive. No, it's not alive. It's a protein. It's a molecule. Where is this happening? Inside a living cell, yeah? So this molecule, it sure looks alive, doesn't it? The fact that you raised your hand thinking it was alive is because you saw it blankety blank and walking. It was walking, right? What made it walk? ATP. Every step I take, you have to pay me an ATP, yeah? ATP is the energy currency, yeah? So it's a very sophisticated machine. There he is, right? There he is walking. Where's the music? Oh, this is another great little video showing kinesin dragging stuff around. He's really cute. This is the Hogengrad lab. So you watch this on your own time. It's really quite cute. And the reason that this is such an important mechanism is because, do you remember how your neurons work? Do you remember synapses releasing neurotransmitters, opening ligand-gated ion channels, allowing sodium to flow through, right? How do those vesicles get all the way down the axon? Somebody's got to schlep, right? All those, and it's kinesin. How long is an axon? How long is a neuron? Can be really blankety blank and long. There's one neuron going from the base of my spine out to the tip of my middle finger, yeah? And why is that? Because the fewer neurons, the faster I can react, right? So there's an evolutionary pressure. If I have a lot of neurons, I'm going to be so much slower, and I won't get away from the saber-toothed tiger that's trying to eat me, right? So having a really long neuron is an essential part of the design, right? Well, how does it work? Again, we know neurons work by releasing neurotransmitters, and those neurotransmitters come from little bubbles inside little vesicles, yeah? You guys remember that? How many people remember this from physio class, yeah? Well, those vesicles are manufactured by the nucleus. The nucleus is a cell body. They're used down at the end, right? Far away from the cell body, yeah? How do they get all the way down there? Well, all along that axon, all inside of it, there's microtubules. And all along those microtubules, there's little kinesin guys schlepping vesicles filled with neurotransmitters so that when it comes time for that saber-toothed tiger to pounce on you, your vesicles are already sitting at the axon terminal ready to go. Does that make sense? So that you can react quickly. And look how dedicated that guy is, right? Where was he? There's the previous
The motor
one.
protein kinesin is a dimer with two.
Right? Look at him. They usually have dramatic music. We need dramatic
for
music.
a little more time to do all the things
Sorry, you guys want to have lunch, I know.
Just choose items from your favorite.
But we need dramatic music. That's the simple fact. Right?
While red
So,
blood cells are carried away at high
this one doesn't
velocity
have
by a
dramatic
strong
music.
blood flow,
I don't want narrated. Yeah, so there's the dramatic music. There's your dramatic music, all right? So these are proteins. P-selectin is a protein that makes a white blood cell bind to a surface. This is your white blood cell, and it sticks to a surface because of its proteins. And there's other proteins that work inside. If we go inside the cell, we can see microtubules being assembled and disassembled. And we have this great music. Sorry, I'll turn it down. And, I mean, look at the job this guy's doing. These organelles rushing it through. Look at his bravery. He's fearless. He's schlepping that organelle. Right? Anyway, so the cytoskeleton is a network on which motor proteins fearlessly and bravely march their way from one part of the cell to another. So these are little highways that eukaryotic cells have. Very sophisticated. Ridiculously sophisticated machinery. Yeah? Okay. All right, and so that is the last part of the first part of chapter 4, part 2. And so